So what is karyotyping? Karyotyping is the process of pairing and ordering of chromosomes after staining. It is done in cells which are arrested in mitotic phase. In these cells, basically the chromosomes are in their most condensed stage. So they reveal a distinct band pattern on staining. And we can identify various structural abnormalities with the chromosomes, like if there are any deletions, inversions, right, that can be identified in the chromosome. Then the chromosome number also can be determined. So here you see it is showing a trisomy of the chromosome 21, which happens in Down syndrome. So this karyotyping helps in identifying various birth defects, genetic defects, and is very useful in research also. Next method for the assessment of function of nucleus is polymerase chain reaction, by which we amplify DNA. So if this DNA is present in a sample, even if in very small quantities, we can amplify into large samples of DNA. So the procedure is we just separate the strands of the DNA and use a primer, and due to the inherent characteristic of DNA to synthesize the other strand, it keeps on synthesizing the other strand. Then the newly synthesized strands are again separated, and again with the use of the primer more DNA is synthesized. So this helps in amplification of the DNA, which may be present in very small amounts in a sample. So it is widely used in diagnosis of diseases like viral diseases. So if DNA is present, and we can actually detect that what DNA is present in the sample by DNA amplification. And not only that, even if RNA is present, we can use RT-PCR for identification of that RNA present in the sample. Then the identification of the amplified DNA is done by blotting. As we said, Southern blotting is done for identification of DNA. So what we do is first run the DNA through electrophoresis, and this method separates the various DNA fragments which may be present in the sample. So we see based on the size, the fragments separate. Then the separated fragments are transferred to a filter membrane so that it immobilizes the separate fragments and they stay there only. Finally, probe is added. So this probe is used based on what DNA we are planning to identify. So suppose if we are suspecting a particular virus, a different probe will be used for the identification of the genome of that particular virus. Okay, then the probe goes and binds to the DNA fragments if that particular DNA fragment is present, and we might add multiple probes also. So multiple probes are added, whichever matches will go and bind with the DNA, and later on with washing we will remove all other probes which are not bound to the DNA fragments, and hence depending on which probe is bound to the DNA fragments, we are able to identify the said DNA fragment in the sample. Now same method is followed for identification of mRNA, proteins, or the post-translation modification of proteins. So similar kind of methods are there, where we add a probe depending on our area of interest, what we want to identify, and then the fragments are identified. Similarly, we can assess function of various other organelles. So there are organelle specific assays based on the enzymes which are present in various organelles. So for lysosomes, we can use the identification of these enzymes like acid phosphatase, beta galactosidase, beta hexosaminidase, lysosome specific dipeptyl peptidase 1. So presence of these enzymes can be assessed for the functioning of the lysosomes. For Golgi we use alpha mannose, then endoplasmic reticulum, G6 phosphatase, that is glucose 6 phosphatase, and for mitochondria we can assess cytochrome C oxidase. So for this, basically we have to break down the cell first so that it releases the various organelles of the cells, and then we have to separate the various organelles. So there is a method in which if we centrifuge the material obtained after breaking down the cell, the various organelles layer into various compartments of the centrifuge. So we can take it out and then assess the presence of various enzymes, and there are diseases in which the expression of these enzymes can be decreased. So with assessment we will know the details of functioning of the various organelles. Then we can also assess the secretory functions of the cells. So we can determine the concentration of hormones, for example. So whether the levels of T3, T4 are normal or not is used to assess the function of the thyroid. Similarly, to find out that why this T3, T4 is less, is it at the level of the thyroid, the disease is there at the level of the thyroid, or at the level of the pituitary. So we can assess the hormones released from the pituitary as well. So this will also give us an indication of various diseases, and then we can assess the specific markers of cell damage. So for example, liver damage is there will be release of aspartate aminotransferase, that is AST, or alanine aminotransferase, that is ALT, into the circulation. So with cell damage there may be certain cellular contents which are released into the circulation. So that also will give us indication of various diseases.